Welcome everybody, I'm Philips, CEO and one of the co-founders of Noscendo. Sepsis is more common than breast cancer, lung cancer, and AIDS combined. And it is a global problem with 27 million people affected by sepsis each year. 9 million survive unaffected, 9 million face lifelong consequences, meaning PTSD, the loss of limbs, or memory and concentration issues, which forces them out of their current jobs. And 9 million people die each year of sepsis. And the reason for that is, or are, inadequate diagnostic procedures to identify the microbial cause. They are non-sensitive and non-specific. Only in 30% of the cases, today's diagnostics reveal the microbe causing the infection and giving the clinician evidence at hand to treat adequately. In 70%, however, the clinician doesn't receive such a result, leaving him with guideline-based treatment, meaning broad-spectrum anti-infectives, and his own experience to judge how to treat. And this is what we change with Noscendo. Using a new class of DNA, which is cell-free DNA, and harnessing next-generation sequencing, our algorithms, our software, our product is able to identify over 1,000 microbes in a single assay from a single blood draw. But not just identifying, we also create relevance towards those findings, meaning we highlight the most important microbes we find and report this to the clinician, creating actionable information towards a treatment which fits best, is most adequate, and most effectively. And all this in a time frame of less than 24 hours compared to days of current standard of care diagnostics. The algorithms we use are validated with samples from 2 different clinical trials, a proof of concept study with 7 patients and 50 samples, a clinical validation study with 50 patients and 250 samples, amounting in clinical evidence of more than 300 samples. And we are right now preparing a multicentric trial with enrolling 500 patients starting next year to create further acceptance and knowledge about the technology and the product. Looking at the results we generated from the second clinical validation trial, we could show that in this trial only 11% of people received positive diagnostic results. 11%. Looking at the same sample, our product could generate positive results in 71% of the cases. Those results were sequentially then evaluated by an expert panel consisting of clinicians and microbiologists, and 96% of those results generated information which was judged as relevant towards treatment. But how does it translate into actual care? That is the main question every diagnostic test has to face. And here is the answer for our product. Every second patient would have been treated differently if our results were available. Every second patient. To give you an example, I would like to introduce Stefan Hofer, who is the head of anesthesiology in a large regional clinic in Germany. He approached us with a problem: I have a really severely ill patient at my ICU. However, all standard diagnostics, blood culture, PCR tests, everything remains negative. Can you help? So we looked into the sample and we identified a virus as the cause of infection. After we reported it to him, he came back and said, "Thank you for the fast results. It was correct, and we adopted treatment based on what you reported." And that's how we help clinicians save lives. So the market we start to operate on is a German severe sepsis and septic shock market with 90,000 cases amounting to roughly 36 million euros of market. Looking at Germany alone, with 300,000 sepsis cases a year, it is a 120 million euro market. Europe, 1 million cases, amounts to 400 million. However, helping all those 27 million cases a year, we are looking at a total market of 2.7 billion euros. This market will be approached by in two ways. First, a send-in model where we orchestrate logistics, sequencing, and diagnostics through the SaaS-based product, which will generate a steady revenue from smaller clinics to the company. And secondly, an enabling model where we bring in protocols and standard operating procedures into the clinics such that we enable the clinics to sequence a sample themselves and just supply the SaaS-based product directly into the clinics. There, the sample never leaves premises. Competition-wise, we're the only one who can distinguish, based on the relevance assessment we do, commensals, contamination from the true infecting agent, while maintaining microbial coverage of bacteria, viruses, fungi, and if necessary, parasites. The team behind it covers all critical aspects, from IT, financing, clinical networks, and laboratory knowledge. Right now, we're raising 2.5 million to get regulatory approval in Europe on the product, to run commercial pilots throughout different kinds of hospitals, ranging from regional hospitals, academic hospitals, into hospital chains located in Germany. We will, as I said, participate in a multi-centric KOL study, and we are expecting to roll out the product first in Germany and then Europe-wide from mid-2018. Having Philips as a partner, for example, with a Papaya platform or with their PCR expertise Philips has, coupled with the bioinformatics IP Noscendo holds, the expertise, the KOL networks, and the knowledge about how to handle a sample, we together can create an integrative diagnostic solution, which is a complete workup of sepsis patients, from rule in, rule out, up to a specific diagnosis, helping patients faster and more effectively. But it won't stop there. The technology we developed and the algorithms we use are able to be also supplied into different fields of infectious diseases, like deep tissue infections, artificial joint infections, infections during pregnancy, and infections occurring in the most precious amongst us, infections in neonates. Thank you.